Good morning, everyone. My name is Magdalena Muskała, and I am a fourth-year student of Medical Analytics at the Medical University of Łódź. I am active in the Medical Biology Research Group. Today, I would like to introduce you to the unusual plant Plectranthus venteri. Perhaps, as the title of our work suggests, it will become the future plant in biotechnology. Plectranthus is one of the largest genera within the Lamiaceae family, with approximately 350 species distributed mainly in Africa, India, Japan, Malaysia, and Australia. Plectranthus venteri is a narrow species endemic to the region of South Africa and was discovered in 1997. It is characterized by light green oval leaves with delicate purple shades on the underside. It is a climbing plant and extremely attractive, also grown as a potted plant. Literature data indicate that this plant is a source of, among other things, bioactive acetophenols and terpenoids. The aim of our work was obtain transgenic Plectranthus venteri roots overexpressing the DXR gene by rhizobium rhizogenes mediated transformation, as well as to estimate the content of secondary metabolites and verify their biological properties in an in vitro model. In this figure, we presented the plant efficiency cassette the XR created based on a vector. The next slide shows the production of hairy roots by rhizobium rhizogenes. Hairy roots are interesting in vitro plant cultures used in basic and applied research. They are very similar to birds' nests. The main advantage of such a system are rapid growth, genetic stability, easy of manipulation, and independence of culture from external conditions. Thanks to them, we are able to obtain larger amount of compounds. We managed to obtain 7 root clones transformed on liquid medium without growth regulators, but only one was used for the research. We confirmed the transformation using PCR. The first channel is negative control. The second is genomic DNA isolated from 2 independent clones. And the third band is a marker of DNA size. In our study, we used methanol extract from transformed roots overexpressing the DXR gene. We checked the cell viability using the MTT assay. As shown in figure 4, Plectranthus venteri root extract reduced the viability of treated breast cancer cells in dose-dependent manner. Based on the cell viability measured after 24 hours, approximate 50% inhibitory concentration was about 1.5 milligrams per milliliter. In this figure, we present the changes in the expression of apoptosis-related genes after 24-hour exposure. Here we use the quality RT-PCR methods. The mRNA level of the anti-apoptotic gene BCL2 decreased, and the levels of the pro-apoptotic genes, for example, BAX or P53, caspase-3 and caspase-9 and apoptotic protease activating factor were increased. In the last graph, we presented that the extract significantly reduced the level of mitochondrial membrane potential in the breast cancer cells studied compared to the controls. This reduction in mitochondrial membrane potential may indicate the apoptotic cascades in cells treated with the extract from transgenic Plectranthus venteri roots. In the next slide, we present a conclusion. For the first time, we were able to obtain overexpression of the XR gene in genetically transformed Plectranthus venteri hairy roots. We demonstrated a decrease in viability of breast cancer cells as determined by the MTT assay. We measured gene expression, for example, pro-apoptotic and anti-apoptotic genes for breast cancer cell line after 24-hour treatment, and we have demonstrated that extract with overexpression of the XR gene significantly reduced the mitochondrial membrane potential level in the tested breast cancer cells compared to controls. Obtaining a highly productive plant in vitro cultures may in the near future be a major source of biologically active compounds with very broad application. Thank you for attention.